My pleasure to start the afternoon with a presentation about the Michelson Found Animals Prize and Grants program. My name is Linda Rhodes. I'm a veterinarian and I have a PhD in endocrinology and physiology, and I've been working in reproduction and endocrinology and drug development for many, many, many years. And I started out with the Found Animals Foundation pretty much from the beginning, about 10 years ago. But it's my pleasure to have Thomas Conlon here with me to help give this presentation. And Thomas is now the Chief Scientific Officer of the Found Animals Foundation. And he has a fascinating background, which you can read about in your Whova app. But in the interest of time, I won't go into all his amazing accomplishments, but we'll launch right into the presentation. He's going to talk about the history of how the grant and prize program came to be, what it's about, what some of the foundational goals of the program are. And then I'm going to go through what we've learned over the last 10 years from the research that has been done by the grantees. And then Thomas is going to come back and talk about the future and some of the ongoing work now that we think is going to lead us into the future. So with that, I'll let Thomas take over, and I'm glad you're all here for the afternoon.
Thank you, Linda. And let me just start by making 2 points. I recently joined y'all's community when I joined the Found Animals as the Chief Scientific Officer, and really appreciate the community that you all have. You are very dedicated. Now that I've been to 3 meetings and seen some of the same people repeatedly throughout, it's really felt that become a part of your family, and became fairly dedicated myself into this goal. And with the background that Linda is going to go into maybe a little bit tomorrow, we feel that we may have more opportunities, or at least new ideas to accomplish this goal. So the Michelson Found Animals Foundation was founded in 2005 by Dr. Gary Michelson, a former neurosurgeon, with the dedication to developing cost-effective, sustainable animal welfare business models to save pets and enrich people's lives. Along with that goal in mind, not only do we have the Michelson Prize and Grants research projects, but there are a number of other initiatives that the foundation is involved in, funding low-cost spay and neuter surgeries, promoting adoption, microchip sales, and a free microchip registry service. There are also 2 adopt-and-shop locations in the LA area where you can shop and adopt animals at the same time. And the latest initiative that the Found Animals has gotten into is the Leap Venture Studio, which is a partnership between the Foundation, Mars Pet Care, and RGA Ventures. It's dedicated as an accelerator program for a pet care industry that's up in upwards of $100 billion a year, to accelerate products, ideas, treatments from sort of the startup stage, and help those companies along developing their ideas. The mission of the foundation: to eliminate the euthanasia of healthy, adoptable companion animals, and to reduce the populations of feral and free-roaming cats and dogs. But more specifically, the MPG program, which was founded in 2008, its mission is to incentivize research through philanthropy and grant funding to develop a non-surgical sterilant for cats and dogs. The prize itself, which we have all heard of, I'm sure, is a $25 million prize for a permanent single-dose non-surgical sterilant for male and female cats and dogs. And there is also $50 million in grant funding available for promising research in pursuit of this goal. The Michelson Prize criteria is fairly stringent, but we'll go into a little more in a couple more slides how you don't necessarily have to meet all of these criteria to be a grantee. But the prize criteria itself requires a single-dose permanent non-surgical sterilant, safe and effective in male and female cats and dogs. It ablates the sex steroids and their effects, suitable for administration in a field setting, that takes into account the feral populations that we have interest in. A viable pathway to the regulatory approval. That means the compound that becomes our product of interest may have predecessors for other uses that have made that regulatory process a little easier, so that we're not necessarily the first group going through with a molecule or a product that hasn't been seen before by the regulatory agencies, and of course a reasonable cost of goods sold in order to make that available for the various populations. The grants themselves, the research is not required to generate results that will meet all the prize criteria. So you don't need to go in with an idea that's going to automatically meet all these criteria if successful. But the approaches need to be a significant improvement on the current technologies. We will fund male-only or female-only projects or ideas. Of course, preference is given to those projects with a potential for resulting in a product that meets the prize criteria. And grantees and non-grantees may apply for the prize. You don't have to have received a grant from the MPG program to apply for the prize. And of course, it is open to academic and industry participants as well. This grant process has brought together people from all facets of science: neuroscientists, oncologists, virologists, chemists, molecular biologists, and of course reproductive physiologists. So we really have attracted, by reaching out to these different populations, these groups of researchers, attracted them a wide variety with different ideas to this granting process. We funded 37 grants over the years, resulting in 22 publications, and $15.5 million have been dedicated to those grants, with $34 million still yet to be granted. We currently have 6 projects ongoing through the grantee process. David Baker at Washington, Patricia Donahoe and David Pepin from Mass General, Christina Gabello at the University of La Plata, David Mooney from Harvard, Benjamin Rehnquist at Arizona, and Lee Smith at the University of Edinburgh. All those projects are currently ongoing, and a number of those groups are here represented as well. And these grantee institutions have really come from all over the United States and internationally: Argentina, Australia, Canada, the Netherlands, New Zealand, and Europe, the United Kingdom. So we've been very successful in attracting ideas from various groups, from different backgrounds, different nationalities, in order to approach this problem. One of the questions we frequently get is, what happens if the proof of concept research from my grant is ready for the next steps, has proved great efficacy, or ready to take it through that regulatory process? And our goal is to see a product that is marketed widely and at a reasonable price, so that we may reach as many populations as possible. These target groups include low-income pet owners, shelters, groups working with the feral populations. And in addition, we will seek licensing and IT rights resulting from these funded projects. That's usually established from the beginning with our contract and granting process. And finally, the Found Animals Foundation, we expect to be involved with the entire process of commercialization through funding and managing the activities. Our scientific advisory board, collectively, we have experience in all these areas of drug development and are well suited to do that, and look forward to that process when the time comes. And at this point, I'll hand it over to Linda to talk about what we have learned so far.
Thank you, Thomas. So I have the enviable position to tell you about all the things that have been done over the past 10 years or so, many of which have not worked. So I get to tell you a little bit about the background, and I think as I was preparing this talk, I was thinking, well, how depressing to go through a whole list of different approaches that haven't worked before. But I think we need to frame it in this way, that at the beginning, there wasn't a lot of money available for research in cat and dog reproduction, as many of you who work in this area know. And there were a couple of ideas that had been looked at over and over again: GnRH vaccines, porcine zonal pellucidate vaccines, GnRH agonists, but there wasn't a lot of new ideas or information. So when the grant process started, we cast a very wide net. We wanted a lot of new ideas, kind of the wackier the better at the beginning, I think, is how we approached it, because we were inviting people into this world that had experience in chemical engineering, that were working on electrospun fibers, or people that had deep roots in human medicine that were oncologists, but they had a way of killing cells that they thought, oh, maybe I can kill oocytes. So we cast a very wide net. We gave a lot of grants to people with really interesting ideas that were very unproven. And the process that we've gone through over the past 10 years is trying to fund the absolute brightest and smartest and most experienced people, giving them their idea and having them prove to us either that it is going to work or it's not going to work. So when we get data from them that's clear and concise, that says, no, this is not an approach that will work, then we can move on. So as you know, in science, it's always useful to get to a go, no-go decision. This is not going to work. I need to do something else. So I'll go through some of the grants, and I'll give you some illustrations of that. But I think it's been a very productive exercise, because now I think the focus of the foundation is really clear on what we think really has potential. So in the early days, lots of ideas. Immunocontraception was kind of on the top of our list. We were interested in targets in the brain, targets in the pituitary gland. We were thinking maybe deslorelin could work if we could get the implant to release deslorelin for long enough. There were some really exciting ideas about how to target specific cells in the gonads, and then there were just some out-of-the-blue ideas that people came up with that we decided to pursue. So we'll start with immunocontraception. And as I said, the zonal pellucidate vaccines had been shown to be effective, and Jay Kirkpatrick's work that was talked about earlier this morning is a good illustration of that. But they're short term, so it's not a single shot forever. Those animals have to be boosted on a regular basis to have an effect. The same thing was true with the GnRH vaccine. The GnRH vaccine that Graham Cox brought on the market for BPH required an initial injection, a booster, and then every 6 months. So those approaches worked, but they didn't work for long enough. So we asked the question, could we figure out a way to get those vaccines to be a single shot? So is there some kind of self-boosting formulation that would present the antigen several times without an additional shot? But we also asked the question, could we look at other targets to immunize? Could we immunize against the FSH receptor, or could we immunize against a sperm protein or something else? So we broke down these grants into two buckets. One was the bucket of different formulations, and one was the bucket of different antigens. So this is the formulation drug delivery. So we've and I'm not going to go through every one of these because there's a lot of grants and it would take us forever. The website of the Michelson Prize Grants has details on each one of these grants on the website. Because one of the wonderful things that Gary Michelson said is we want to make sure that negative data is available so that those things that haven't worked won't be tried over and over again by other researchers. So we looked at biodegradable beads that could regulate the antigen coming off of the implant, coming off of the injection. We looked at David Putnam from Cornell had this interesting idea that he could somehow get a self-boosting formulation that using these microparticles that would keep the antigen in the injection site, and then at a certain point they would degrade and release antigen, so you would get a second boost. Michael Monks thought that perhaps we could do a viral vector to deliver the antigen. And there's a grant now still in progress from Harvard looking at what they call infection mimicking biomaterial scaffolds. So the idea is not just to stimulate an antibody response, but also to stimulate a cellular-based immune response that might be longer lasting. Not counting David Mooney's work, none of these formulation approaches actually were effective in increasing the length of the immune response. We had 4 or 5 different groups look at novel antigens, sperm antigens, different zona pellucida glycoproteins, and then multiple copies of GnRH that could potentially serve as a more robust antigen. And really the conclusion we came to was that the barriers to immunocontraception are too high at the moment to be able to overcome. The single shot, we have not been able to find a formulation that works to give a lifetime immunity. So currently the Michelson Prize Grants is moving away from immunocontraception unless an investigator has some preliminary data that can really indicate a much longer term immunity. We just haven't seen that technology yet. So this is on our list of we tried with really sophisticated approaches and we were not able to overcome this barrier. So moving on to the brain. So what are we looking at in the brain? I love this dog brain, particularly the cat garbage one. I think that's a really important part of the dog brain. GnRH neurons are in the brain, but they have projections that go down to outside the blood-brain barrier. So we thought perhaps we could find some way of killing or inactivating those neurons, because that would truly be the master control. So if you could suppress GnRH neurons, you could suppress reproduction in males and females, and in dogs and cats, because GnRH is a common hormone across species. So it's a great target, but it's a hard target. And as you look at some of these universities and institutions where we funded this research, we tried to choose really the best of the best to go after this target. And there was some real interest in trying to ablate the neurons using a kisspeptin matched with a toxin, that the idea would be the kisspeptin would hone into the receptors on these GnRH neurons and the toxin would be delivered and we could kill those cells. The whole idea of a targeted peptide or toxin that could go directly to those cells had been explored in several different ways. Also a kisspeptin antagonist. And what we found is that these molecules would bind the GnRH neurons, and they could actually, even in the case of the work done by Scott Struthers at Crinetics, they could cause an LH surge, but they didn't kill the neurons. There just was not a delivery of that toxin in a long enough and an effective enough way to ablate those cells. We also had a couple of investigators looking at RNA interference, thinking that we could deliver interfering RNA directly to those GnRH neurons in the hypothalamus. We had problems getting that construct into the brain. And then we thought, well, maybe we could develop vectors that would go specifically to the hypothalamus, but we were not successful in demonstrating that kind of targeting. So in summary, the brain targets, the blood-brain barrier is an issue, even though those projections of the GnRH neurons are outside the blood-brain barrier. You still have to get retrograde transport into the brain, or you have to get it into the brain through the blood-brain barrier. And also, probably most of the neurons have to be killed in order for this to be effective. There's some research that indicates that unless you ablate 90 to 95% of those neurons, you can still support reproduction. So that is a very, very hard target, a very high target to overcome. So the brain, killing those brain neurons does not seem to be a useful approach. The next one is one that's been kicked around for many, many years. It's the same idea of targeting a toxin to a cell that's important in reproduction. So in this case, it's targeting a toxin using GnRH to target the pituitary gonadotroph. So the idea is the pituitary has all these receptors for GnRH on its surface. If you take GnRH and put it with a toxin, it will hone into those pituitary gonadotrophs, the toxin will be incorporated, it will kill specifically those cells, and then you can ablate reproduction. There were Terry Nett's group from University of Colorado first pioneered this work and did a lot of work, and some in dogs, and never was really able to show a single shot long-term suppression of reproduction. So several grants were funded looking at this approach, looking at a way to slow release GnRH to ablate those gonadotrophs. It's kind of an immunocontraception approach, but with the pituitary as a target. Scott Struthers from Crinetics again said, well, if I can't do the brain, maybe I can do the pituitary, and he did a lot of work with this GnRH toxin conjugate, using different toxins, using different formulations, and he really couldn't cause a suppression of reproduction. And now Dr. Ben Redenquist is doing this work with another construct, and he felt that part of what was happening was the toxin was getting into the cells, but it was being sequestered in exosomes. So the toxin was there, it was delivered properly, but it wasn't effective in killing the cell. So he's working on some compounds that also will inactivate those exosomes to enable that toxin to be delivered. So that's in progress. So the summary of the barriers for the gonadotrophs are that they seem to be pretty resistant to toxin effects. A single exposure to these potent toxins does not seem to kill these cells. Perhaps the receptors on the cells have been downregulated so that there's just not that many receptors to accept that toxin. It looks like the toxin can be inactivated by these cells, and perhaps you just need a longer exposure than a single injection. So this is a much harder target than we initially thought 10 years ago. So deslorelin, we know that deslorelin works. We've got a 6-month implant. We've got a 12-month implant. It seems intuitive that you should be able to take enough deslorelin to put it in an implant to get a 10-year implant, which would essentially be a reproductive lifetime of cats and dogs. In human medicine, GnRH agonists have been incorporated into implants that last as much as 1 year, but nobody has ever really cracked that 1-year implant barrier. So we thought we would look at the possibility of extending the life of a deslorelin implant. So Dr. Lanuti at Ohio State had explored this electrospun nanofiber formulation that he felt could slow release over a much longer period of time. But when he did that work, he did not get enough of a release of deslorelin from those implants to cause a suppression of reproduction. And the technology, the implant technology, has really not been shown to be sufficient enough to go for such a long period of time. You'll hear about Dr. Christina Gabella's work tomorrow, but she had a very interesting idea to put these implants in very early, because very early in the reproductive cycle of puppies and kittens, there's an increase in GnRH, and the idea was perhaps if you could suppress that initial increase, you could basically inhibit reproduction for the lifetime of the animal. But her work very clearly showed that you could get a nice decrease, lengthening of the time till puberty, so the animals would come into puberty much later, but they still would eventually be reproductively capable. So that was our approach to deslorelin, and we just don't think that there is a technology on the market that can release it over a 10-year period. So this is all bad news. And there are a few projects ongoing that I've talked about, but generally I think we've been ruling things out. If you target the gonads, you're much less likely than if you target the brain or the pituitary to be effective in both sexes. But as Thomas said, the Michelson Prize Grants folks are certainly willing to fund this kind of research because they understand that an effective all-female contraceptive would be a very useful tool for us. So a number of grants were funded in this area because there certainly are a lot of targets in the gonads. I won't go through each one of these in detail, but I think you can see from the names of the institutions where these people were working. These were really serious researchers who were trying to target these new targets. And so far, for a variety of reasons, none of these have proved effective to be a single-shot, long-term approach. Here's another list: University of Pennsylvania, Virginia, University of Edinburgh. The Lee Smith grant is ongoing, and there are some fairly promising data from his laboratory. But most of these approaches are technically really hard to do. It's hard to get whatever you're administering with a single injection to go directly to the gonads and have a suppressive effect. So the barriers are really, as I said, one sex only. A lot of unproven targets, so you can identify proteins in sperm or in oocytes that could be potentially specific to those tissues, but aren't really proven out as a target. And if you identify an egg protein, for example, you may have some of that protein in the kidney, or you may have some in the brain, or you may have some somewhere else, so you get an off-target effect of your treatment. So many of these targets are early on unproven, and the specificity is really challenging. So the novel approaches, and I just list a couple of them here. Dr. Lamar at University of Guelph was targeting a special kind of RNA and that pathways that were important in meiosis. And so his idea was, you don't have to kill cells. If you just inhibit meiosis, the animals won't be reproductive. The females will not be able to produce viable fetuses. And then Dr. Bentley was interested in the gonadotropin inhibitory hormone, which had been originally, I think, identified in poultry, not really well characterized in mammals. But neither one of these approaches progressed far enough to actually want us to take them to the next step. So I think that the take-home message of this is that the outreach of the grant program has been fairly wide, and I know I've rushed through these grants. A lot more detail is available on the website, but I wanted you to really have two take-homes from this overview. One is that a lot of work has been done by a lot of extremely experienced and innovative researchers. And two is we've learned a lot of what is not going to work and what we don't want to fund anymore and what we think is not going to be the direction. A lot of very, very interesting biological insights have come out of this work. As Tom said, there's been more than 22 peer-reviewed publications come out of this work. So certainly we learned a lot, but in terms of tackling our core need, we need something different. So I'm going to turn this over to Thomas, and he's going to be talking about the next steps.
Thank you, Linda. So to give a little background, I spent 19 years in the arena of gene transfer for treating human diseases. And I had the opportunity to meet the representative from Found Animals at one of our annual meetings a number of years ago. One thing led to another, and they started receiving more applications or ideas based on gene transfer technologies, and was invited back to join the board with Linda and the other fine folks here. And now I'm in a bigger role because of our emphasis on these new technologies I'm going to talk about. Gene delivery is usually based on a viral vector. It doesn't have to be a viral vector. It can be what we call naked DNA. It can be DNA that's encapsulated in a lipid that also gets into the injectable into the body. But primarily, we're focused, and my background focuses on using a viral vector to deliver a relevant gene. And again, that gene doesn't necessarily need to be a gene like GnRH or kisspeptin or an antibody or an antigen. It can be a short piece of DNA, such as an siRNA that silences the gene. And these technologies have resulted in a lifetime expression in a variety of cases, especially animal models of human diseases and companion animals themselves, which I'll show you a little bit about. The question is, can we get a lifetime effectiveness for contraception with a single shot? And I'm going to focus on a typical gene transfer virus that's called adeno-associated virus, AAV. It's a naturally occurring virus. 90% of us in here have been infected with AAV, but it does not cause disease. We know you've been affected because we can look for antibodies to the virus. It's a very small virus, about 5,000 base pairs long. We have billions, 3 billion in our bodies. It's single-stranded. And recombinant AAV, we actually remove all of the viral genes, of which there is only 8 in this virus. So it contains none of the genes that it naturally has. Other than 300 base pairs, AGTC, on either end, everything else is removed, and we put in the DNA that we want to use, including the gene we want to express. AAV is very good at establishing itself in tissues for the long term. One of the questions or problems that usually comes up is people ask, does it integrate into the genome? Recombinant AAV has the potential to do that, but it does that on very rare cases. There are other gene therapy viral or gene transfer viral vectors that the purpose is to integrate into the human genome. And that depends on, that's disease-specific application. So we've shown over the years that these viral vectors can establish themselves for long-term within specific tissues. And there are, just like there's hepatitis A, B, and C, there is AAV 1 through 100 plus at this point, naturally occurring variants. And each of these variants has propensity to go to certain tissues, such as AAV2 and AAV8, likes infecting the liver or muscle. AAV5 is great at transducing the lung. AAV9 is great for transducing the heart and the brain, as well as the liver. So as Linda mentioned, the different targets, tissue targets, brain, pituitary, gonads that are some of our targets, there is potentially AAV viral vectors that go to the tissues of interest where we would want to express a gene. In addition to the naturally occurring versions, there are tens of thousands of man-made versions now that people are making in the lab. It's very easy to do what we call directed evolution in order to create a virus that specifically goes to the tissue of interest, whether it be the kidney, bone marrow cells, etc. So we're not limited to just this panel here. In fact, one of our grantees did focus on targeting AAV to the hypothalamus as well. So gene transfer, you've heard in the last year, 2 products have been officially approved by the US FDA now. One is for blindness and one is for a form of cancer. But there are hundreds of clinical trials ongoing, not just with AAV, but other viral vectors such as lentivirus. You may know it better as HIV. And in fact, because it does infect bone marrow cells and hematopoietic stem cells very well, it's a great viral vector to use for diseases such as severe combined immunodeficiency. But we call it lentivirus just so that we don't use the word HIV out there. And so there's a lot of experience with this technology, and it's growing constantly. And in fact, it's being used to treat companion animal diseases as well, and has for quite some time. One of the first studies I was involved with at the University of Florida was for dilated cardiomyopathy in Doberman pinschers. But in addition to that, people are researching expressing a gene anti-myostatin just to treat old age and muscle weakness in companion animals, congenital achromatopsia in dogs, which would be using the blue cone opsin gene to treat a form of blindness. There's some for cancers, as well as GT, which occurs in the Great Pyrenees That's using a lentiviral vectors that we just talked about. So there's experience with developing products and treatments in dogs and cats as well as humans with these technologies. In fact, in regards to gene delivery for contraceptive, there have already been 2 patent applications that have been submitted by Dr. Wilson at UPenn and Dr. Hay at Caltech, and published peer-reviewed research by Dr. Lee and his group expressing an antibody from AAV for long-term contraception. So the idea is out there, and it's not just this group or MPG that's funding these ideas, but others are also working on these projects as well as potential products that they could follow up with as well. An example of a project we're working on that uses gene delivery is delivering the malaria inhibiting substance via an AAV vector to block follicular development. That's just one example of what viral deliveries could be used for. And Dr. Pepin, who's here, is going to talk about this study itself tomorrow and more in depth. But you can imagine with gene delivery approaches, you could deliver antibodies, you could deliver antigens. You know, Linda just talked about how, you know, we have this Lorezin for 6 months or 12 months, but we just can't get that that immune response to keep kicking out the antibodies longer term, up to 10 years is what we're looking for. Perhaps we could use gene delivery to express an antigen for longer and thus initiate that immune response over the years. So there's lots of potentials out there, including silencing as well. So there's proof positive out there that gene delivery offers potential for a single shot lifetime effectiveness. And the progress in human medicine that's occurring and is growing exponentially in the last couple of years is going to pave the way significantly from the regulatory process, from the manufacturing process, which is quite an issue in gene transfer technologies right now, but it is growing daily. And what I had the opportunity when I was at the University of Florida, and by the way, I did work with Julie Levy a little bit, and I was just reminding Linda a little earlier that I worked with John Verstegen before he left the University of Florida as well, so I had a connection quite a while ago. And when I was at the University of Florida, we of course worked on human diseases, and we had a group come to us from Taiwan. Because they have a small island population, a gene for adenosine decarboxylase deficiency sort of stayed in that island population. There are a few patients outside of there, but there was a high rate of incidence within the Taiwanese population. Being a small country, they didn't have the technology that they needed. They knew what gene was missing, but they didn't have the know-how and manpower to get a therapy developed, made, etc. So I was involved with getting the approval through the Taiwanese government, which their version of the FDA is called the CDE, to get that approved so that they could do a compassionate use protocol. These patients with this deficiency usually pass between 2 to 3 years of age. It was an awesome experience. We did get approval. We were able to treat these kids. A compassionate use protocol is a protocol that's not a formal clinical trial. This is their only chance, and they know there's no other option. So, and at the time, actually, the U.S. didn't have compassionate use protocols for gene transfer at the time. Now they do, and there's actually been 3 that have been initiated in single patient trials. So what I wanted to show you was the impact that gene transfer can have on human patients and the potential impact that it could have for our companion animal populations and feral populations as well.
怒ったことだっていっぱいあるから。
わあ、3人目だっておっしゃられて。
おお、来た来た来た来た。おお、できた。恥ずかしいって。それ可愛いね。上手。
寝たきりだった子どもたちが歩き始めた。日本でたった3人しかいない希少難病の子どもたち。明日を生きる希望をあなたに。
But it's, I was going to say that they have an official clinical trial ongoing right now that you can, of course, look for additional videos as well as these individuals that were included in the compassionate use protocol. And to update, some of them are learning to walk now. So, you know, they wouldn't be around at this point without this treatment, and it's really neat to see. So hopefully we can have that same effect for our populations, animal populations as well, in the way that we want to. Some of these patients are now out 6 or 7 years. In some cases, that's the reproductive lifetime of a cat or dog, from what I understand. So there's proof positive there as well that you can get gene expression for the long term.
We'll take questions.
Happy to take any questions at this time. Both of us will. Thank you.
Michelle Kutzler, Oregon State University. Thank you for that presentation. I have 2 questions, but I'm going to make it really fast. Whether founded or not, the public has concerns about genetically modified organisms used in the manufacturing or creation of human food. I foresee this as a major concern for extending the scalable efforts for dog and cat sterilization that targets or uses gene therapy to destroy reproductive tissues. And I'd like to not poo-poo the idea of vaccines. I know that there have been some limitations, but I was very excited about a lecture that I heard back in 2000 at the International Congress on Animal Reproduction by Dr. Richard Farrer-Hoskin. In fact, I pulled up the abstract on my phone just so I could read it to you. He said briefly, population control in dogs and cats is an important goal. I'm just going to skip down to the bottom. Specifically, vaccination with purified ZP glycoproteins leads to initially immunocontraception and then profound irreversible changes to immunosterilization. So he was vaccinating dogs, not cats, and we realized that obviously they're different, and that's for another subject altogether. But what he reported, and unfortunately was not included in his abstract, and I know I've brought this up at this meeting and with many of you before, was that he vaccinated young dogs, he vaccinated pregnant, I should specify, female dogs, and what he found was not only did they stop, technically what he found was that they essentially developed immune-mediated oophorytis, so they had large unovulated follicles, which then completely removed all of the oocytes from their ovaries. And so he had sectional images of essentially no follicles present at all in their ovaries. We did a study with the PZP vaccine in horses and had some similar results. So I mean, these were not temporarily sterilized. They were permanently sterilized. They didn't have to maintain antibodies for the rest of their life because they no longer had follicles. They no longer had ovaries or oocytes. Unfortunately, I don't know whatever happened to that. He said that it was the Ovax vaccine. Of course, I can't find anything like that in the literature. And I think that there's certainly an opportunity to consider looking at that again in dogs. But that was my question, quote unquote.
Let Linda address that question. To go, your first statement was in regards to the potential for gene transfer treated animals to be considered GMOs. That's definitely something we've been considering on the board and talking about for some time as we've developed more into these technologies. Linda's been integral in informing me the various thought processes or thought groups that are out there that may have different opinions about what these animals would ultimately be termed and considered by the population in various groups. So I really appreciate you expressing, and I would love to talk to you more about your feelings and the direction that if one of these products became reality, what direction you would take in helping people accept it or understand it in better ways. So, but that time will have to come when we're ready. Yeah.
Yeah, it's a great point, Michelle, and I think you'll get from, if you look at the program, you'll get a sense that the Alliance for Contraception in Cats and Dogs has acknowledged that could potentially be an issue, and so we've structured this conference to start introducing gene delivery technology to this audience and hopefully more broadly, and we've invited Ricky Lewis tonight to talk to us about its applications in human medicine, so that people, I think, begin to get familiar with what the technology is and not be so scared of it. So thank you for that. Joanne.
Yes, it's on. Can you hear me? Let me take this out. Is that better?
Yeah.
I'm Joanne Mackey. I currently work for Boehringer Ingelheim, who used to be Marielle. I've been involved with ACC&D now as a technical advisor, and these are my personal opinions. I do admire the Michelson Prize because it really brought this issue to light. If you hadn't done that, I don't think this whole focus on really getting to the problem and getting something right. What concerns me a bit as a sterilant is if a company was going to be looking at an immunocontraceptive for cats and dogs, the word sterilant concerns us because we know in our current products there's a lot of potential misuse or off-label use, and the idea that a sterilant would be out there, it might possibly be applied to other species other than cats and dogs. With that statement being made, if you had a product that wasn't permanent but could be administered in a way, say every year or every few years with rabies vaccine campaigns in the field, it would be driving people to those rabies campaigns and would also then potentially promote good health care and getting that dog or cat to have an owner associated for long term. So I admire the goals. I think they're worthy goals, but I'd hate to lose sight of a fact that there could be an excellent use of an immunocontraceptive if you can find one that can make the 3 to 5 year length.
Yeah, Joanne, I think that's a great point. I think one of the important things to remember as we do all this research is that we've talked about this for a long time, but we would like to see a creation of a toolbox of approaches. So we love Deslorelin as a 6-month and 1 year implant. It's a new tool, it's a relatively new tool, it's terrific. It doesn't solve the feral cat pet population control problem. We need something different for that population. A 3 to 5 year immunocontraceptive might be extremely useful in the control of feral horses. So I think we have to think of it more broadly in not a single silver bullet, but a toolbox of tools that are going to be useful in a lot of different situations. And I think the focus of Gary Michelson on specifically a lifetime is driving this whole research forward. But it doesn't mean that if we find a 3-year product, it's not going to be useful. And one of the things I'd like to promote is the fact that ACC&D has been very curious about how long is long enough. And so we started quite some time ago working with Dr. John Boone to develop very sophisticated computer models of population controls of feral cats to ask the question, is 3 years enough? Is 3 years as good as surgery, or does it need to be 4 years, or does it need to be 5 years? How much of the population do you have to contracept? And actually asking those questions in a very rigorous and science-driven way. So I totally take your point that we're not just looking for one solution. We're looking for a handful of solutions that will be used in different ways. I think it's a really important point. Thank you, Joanne.
I'm afraid we have to cut off questions now, but they will both be around the rest of the conference and tonight. And I want to thank both of you for both this good presentation and your commitment, longstanding, to what is a difficult challenge, but one that there's a lot of progress being made. So, hand for Linda and Thomas.